for gluconeogenesis. Gluconeogenesis is the opposite of glycolysis. This is when you're using the pyruvate to make glucose. It usually happens in fasting states. In fasting states, the insulin basically decreases and glucagon increases, and the body needs some energy for the RBCs and especially the brain to go on. One more thing is that there are three molecules that can ultimately be used to create glucose. One of them, you can remember them by a mnemonic called LAG. One of them is lactate. Lactate gets converted to pyruvate. Alanine also gets converted to pyruvate. This is your second molecule. And the third molecule is glycerol, right? So the actual even chain fatty acids cannot make glucose, but the glycerol backbone can. Like the glycerol backbone from the triglycerides can enter gluconeogenesis and make glucose. The glycerol basically can gets converted into DHAP, right? Can you see this DHAP here in gluconeogenesis? So this is where the glycerol enters, right? Okay. So even chain fatty acids cannot make glucose, but the glycerol backbone of triglycerides can enter gluconeogenesis, and all chain fatty acids are the exception, right? They can enter the all chain fatty acids can enter gluconeogenesis. Now the first step is the pyruvate, right? So the pyruvate needs to be converted into most of the steps of of glycolysis are reversible. So the reversible steps are not a problem, right? So this is reversible, this is reversible, this is reversible, this is reversible. But some of them are not reversible, right? So for example, this is not reversible, this is not reversible, and this is not reversible, right? So the irreversible steps use special enzymes to go the opposite way around. So there are only four specific enzymes you need to know for gluconeogenesis. The rest are the same for glycolysis, right? So let's start. So glycolysis was basically coming from glucose to pyruvate. Now from going from pyruvate to glucose is gluconeogenesis, right? The first step is to take the pyruvate and convert it into phosphoenol pyruvate. Now this is done in two steps. First, we take the pyruvate and convert it into oxaloacetate, and this is done by pyruvate carboxylase. This is your first reaction, right? So you want to go from the pyruvate to the phosphoenol pyruvate, and this road is one way. The liver takes a detour, right? So the liver basically takes the pyruvate, converts it into oxaloacetate, and this oxaloacetate, by the use of PEP carboxykinase, which is basically phosphoenol pyruvate carboxykinase, gets converted into phosphoenol pyruvate. Now, this step is important because pyruvate carboxylase, it needs ATP and biotin, right? And this happens in the mitochondria. So pyruvate basically needs pyruvate carboxylase, it needs ATP and it needs biotin and gets converted into oxaloacetate, and only then can it go into the gluconeogenesis pathway. This is very importantly tested, so I'm going to punch it here. Can you see that the pyruvate carboxylase basically needs ATP and biotin, which is your vitamin B7. So biotin deficiency will also lead to a problem with the gluconeogenesis pathway. Why would the body activate this enzyme? So here's the beautiful connection. When you're fasting, your adipose tissue is releasing fatty acids. The liver burns those fatty acids by beta oxidation. That produces a lot of ATP and acetyl-CoA. When acetyl-CoA becomes high, it activates pyruvate carboxylase. So acetyl-CoA basically tells the liver we're burning fat for energy, send pyruvate down towards making glucose. Right? So what are we doing? We're increasing acetyl-CoA, we're increasing pyruvate carboxylase, and that pyruvate carboxylase is actually making gluconeogenesis. Right? This is a very, very important connection here. So I am going to punch this down and we're going to go through this again because sometimes reading makes more sense than listening, right? Here we go. So the situation is that you're fasting, right? And your adipose tissue is releasing fatty acids. Liver burns those fatty acids by beta oxidation, and that produces a lot of ATP and acetyl-CoA. When acetyl-CoA becomes high, it activates pyruvate carboxylase. So acetyl-CoA basically tells the liver, we're burning fat for energy, send pyruvate towards making glucose. Therefore, acetyl-CoA increases the pyruvate carboxylase, and that increases the gluconeogenesis. Right? Okay, moving on. We move on to the next step. Okay, so now we have pyruvate. Now we have pyruvate that is converted into oxaloacetate, and now we're creating phosphoenol pyruvate using the phosphoenol pyruvate carboxykinase, right? So phosphoenol pyruvate carboxykinase, this step uses GTP, right? So memorize this little pair. The pyruvate is converted into oxaloacetate using pyruvate carboxylase, right? And the pyruvate carboxylase basically means biotin and ATP. And the oxaloacetate is converted into phosphoenol pyruvate using the PEP carboxykinase, right? That's the hardest part to remember of this entire reaction. If you remember that, everything is good. I'm just going to make an arrow here to make you remember that PEP carboxykinase basically uses GTP because this is tested. It uses GTP, right? So this is the most important tested part. Okay, so you just take pyruvate and through pyruvate carboxylase, which was activated when you're fasting, because the adipose tissue is releasing fatty acid, the liver burns this fatty acid by beta oxidation. That produces lots of ATP and acetyl-CoA. When acetyl-CoA becomes high, it activates pyruvate carboxylase. So acetyl-CoA basically tells the liver we're burning fat for energy. Send pyruvate down towards using glucose. Therefore, acetyl-CoA basically increases pyruvate carboxylase. And this is when you're fasting, the gluconeogenesis increases, right? So this is upregulated, this pyruvate carboxylase is upregulated in fasting when the fat is being broken down, right? And the oxaloacetate is basically using the PEP carboxykinase into to create the phosphoenol pyruvate, right? I'm doing this again and again because this is the only part that is actually tested when it comes to gluconeogenesis, right? Now things become easy. Once basically you create the phosphoenol pyruvate, the same steps go in the opposite direction. You're creating the 2-phosphoglycerate using the enolase, you're creating the 3-phosphoglycerate using phosphoglycerate mutase, you're creating the 1,2-bisphosphoglycerate using the phosphoglycerate kinase, and the glyceraldehyde 3-phosphate using the glyceraldehyde phosphate dehydrogenase, and then you're using triose phosphate isomerase to create the DHAP. And by the way, the glycerol was directly being converted into DHAP and entering here, and then the aldolase was converting it into fructose 1,6 bisphosphate. Now another irreversible step happens, right? So the next one-way street happens when you reach the fructose 1,6 bisphosphate. The fructose 1,6 bisphosphate goes on to make the fructose 6 phosphate by this special enzyme called fructose 1,6 bisphosphatase. Now, this fructose 1,6 bisphosphatase, this enzyme is very, very important. Why? Because it's one of the major regulatory points deciding are we burning glucose or making glucose. So this is where we're deciding are we making glucose or are we burning glucose. If we're burning glucose, we're using phosphorotockinase. If we're making glucose, we're using fructose 1,6 bisphosphatase. So here's the logic. Suppose the cell has lots of AMP, right? What does AMP mean? AMP means low energy. Would this be a good time for the liver to spend huge amounts of ATP making glucose? No. So AMP inhibits fructose 1,6 bisphosphatase, right? So in basically low energy states, when you have a lot of AMP, there's not enough energy, the body will not waste the ATP in creating more glucose. So you can just, I'm going to punch it here. You can arrow and get my text box working here. So basically, AMP inhibits this, right? Now, therefore, a lot of AMP will inhibit gluconeogenesis, right? That makes sense. Then we continue upwards. The fructose 6 phosphate, right, becomes a glucose 6 phosphate, right? This is a reversible step. It's done by hexose isomerase. And then we have to come to the last step. The last step is again the last one-way street, right? So in glycolysis, what happens is glucose becomes glucose 6-phosphate using the hexokinase, right? And this is an irreversible step. This basically commits the glucose to glycolysis, as I told you. Now, the glucose 6 phosphate needs to be converted into glucose, and this is glucose 6 phosphatase. To do this, we need to free the glucose. Now, the liver releases that glucose into the blood, and once the glucose is freed, your mission is accomplished. So, the whole pathway is basically this, right? So, glucose 6 phosphatase is very important, right? So there are four special enzymes, right? Which four special enzymes did we study? The pyruvate carboxylase, the PEP carboxykinase, which uses GTP. The pyruvate carboxylase uses biotin and ATP. Then we had the fructose 1,6 bisphosphatase, which AMP basically inhibits. And then we had the glucose 6 phosphatase. So now let's understand glucagon. This becomes incredibly simple if you remember the body's intention. Now you haven't eaten. You're in a fasting state, therefore glucagon increases. When glucagon increases, what does glucagon want? It tells the liver, put some glucose into the blood, there's no glucose. So glucagon increases glycogenolysis. Basically, it breaks the glycogen into more glucose, and it also increases gluconeogenesis. It tells the body to make glucose while decreasing hepatic glycolysis. So basically, it tells the liver to not break down more glucose. Okay, so I'm going to punch this text down here, and I'll have you guys like pause this video at this point and just read this and try to understand if it makes sense. Okay, now because it would be ridiculous at this point, can you imagine that it would be very ridiculous for the liver to make more glucose and immediately burn glucose? Like it would make no sense during fasting. So during fasting, the liver shifts toward making glucose and not burning it. Now the famous there's a famous fructose 2,6 bisphosphatase thing, right? There's a famous fructose 2,6 bisphosphate thing going on here. This looks horrible in books, but the idea is very simple. There's a molecule called fructose 2,6 bisphosphate. Phosphate. Its job is basically to push the liver towards glycolysis, right? Let me just punch this down here so you guys can refer to something while I'm talking about it, right? Okay. So let me punch this here. Okay. So there is a special molecule called fructose 2,6 bisphosphate, right? Its job is basically to push the liver towards glycolysis. So its main job is to tell the liver to break down glucose and you make ATP, right? Therefore, fructose 2,6 bisphosphate is high when you're basically doing a lot of glycolysis and you're not doing gluconeogenesis. Now, the glucagon wants the opposite. Whenever your glucagon is high, it basically wants gluconeogenesis to happen. So, when glucagon basically, whenever there's glucagon in the blood or the body, basically, you have a decrease in fructose 2,6 bisphosphate because fructose 2,6 bisphosphate, when it's decreased, obviously it will lead to less glycolysis and increase gluconeogenesis. So glucagon and fructose 2,6 bisphosphate are like anti-molecules. Whenever one is high, the other one is low. Okay, this is the only thing that the examiners want you to remember. And insulin goes hand in hand with fructose 2,6 bisphosphate because insulin wants the same things that fructose 2,6 bisphosphate wants. Right? This is not that confusing, guys. If you go through this slowly, read these things, try to understand them, it makes sense. Okay, now some clinical stuff basically. The clinical stuff is that this is where examiners ask you a lot of questions. In alcoholics and fasting, what do you get? You get hypoglycemia. So suppose someone hasn't eaten all day, their liver needs gluconeogenesis to maintain the glucose, and they drink a lot of alcohol. So ethanol metabolism produces lots of NADH, and high NADH causes the pyruvate to get converted into lactate, right? And the oxaloacetate gets converted into malate, right? So we remember our um, TCA cycle where the oxaloacetate was getting converted into malate, right? Now, wait, we desperately needed pyruvate, right? I think it will be difficult for you guys to follow this case, so I'm going to write it down here. Okay, so alcohol and fasting. Let me copy it down here. Okay, excellent. Put this here. Very nice. Okay, let's try to understand this. Now, when you have alcohol and fasting, you get hypoglycemia, right? Suppose someone hasn't eaten all day, their liver needs gluconeogenesis to maintain glucose. Then they drink a lot of alcohol, ethanol metabolism produces lots of NADH, right? And high NADH causes a lot of pyruvate to get converted into lactate, right? Now, a lot of pyruvate is converted into lactate. and a lot of oxaloacetate is converted into malate, right? Now wait, we desperately needed pyruvate to get converted into oxaloacetate so we can make more glucose, but that's not happening here. Alcohol is pulling away both the pyruvate and the oxaloacetate. So the opposite thing to TCA is happening here, and the pyruvate is not going into acetyl-CoA, right? So alcohol is literally pulling away both pyruvate and oxaloacetate. So what happens is in alcohol poisoning or the alcohol over the drunks, what happens in them is the NADH increases. The pyruvate becomes lactate, right? And oxaloacetate becomes malate. The opposite thing is happening in TCA. So in TCA cycle, normally the malate should become oxaloacetate, but here the opposite is happening. And the pyruvate is not going and becoming acetyl-CoA to get going into the TCA. It's literally becoming lactate, right? It's making the situation even worse, right? Normally, the pyruvate should go and convert itself into acetyl CoA so we can use it in the citric acid cycle. But what alcohol does it? Alcohol takes the pyruvate and converts it into what? It converts it into lactate, right? And that basically also produces your NAD plus. So you get a lot of NADH in alcoholics, you get the pyruvate becoming lactate, you get the oxaloacetate becoming malate, and you get a decrease in gluconeogenesis. You get a decrease in gluconeogenesis because how will you how will you start off with glucogenesis when the pyruvate is quickly being converted into oxaloacetate, right? Now that leads to hypoglycemia because the pyruvate is becoming lactate, and this is why you get lactic acidosis in alcoholics. I really hope that was easy to understand. I really tried to make you guys understand it. A classic question would be a malnourished person who drinks heavily without eating and is found to be confused and sweaty, and the labs will reveal a reduced glucose and an increased lactate. Why? Because excesses NADH, a lot of NADH basically inhibits gluconeogenesis, right? That is very, very important for you to know. Always remember, um, let me punch the text button here. This is very, very high yield. It's very commonly asked on exam questions. So, malnourished person drinks heavily without eating and sound confused and sweaty. Labs will reveal a decreased glucose and increased lactate. Why? Because excess NADH inhibits gluconeogenesis, and this is very, very, very high yield, right? Okay. Now we have some diseases that we need to study, and the first one is the von Gerke disease, right? Whenever this glucose 6 phosphatase, this enzyme, whenever it's missing, we call this disease basically the von Gerke disease, right? And it's very commonly questioned asked. This is the von Gerke disease, right? This is when this enzyme is basically absent, right? So imagine this enzyme is absent, the glucose 6 phosphatase is absent. So you can't really make glucose. The liver cannot properly release free glucose, right? What happens is there is severe fasting hypoglycemia, right? There's no new glucose can be released by the liver, and the liver gets packed with glycogen metabolites, and this leads to hepatomegaly. There's also increased lactate. Um, there is also increased uric acid. There is increased triglycerides and lipids. So if you see a child with hepatomegaly, severe fasting hypoglycemia, and lactic acidosis, you always think about von Gerke disease. Now, why am I constantly punching these things here? Because these are very, very commonly asked, right? So, you don't have the glucose 6 phosphatase, you can't release the free glucose, right? You get severe fasting hypoglycemia. This is called von Wierke disease, and the liver gets packed with glycogen metabolites, and this leads to hepatomegaly. So the lactate rises, the uric acid rises, triglycerides and lipids rise. So if you see a child with hepatomegaly, severe fasting hypoglycemia, you think about and lactic acidosis, you think about von Wierke. Right. Another one beautiful muscle question could be like, why can't muscle give its glycogen derived glucose to the rest of the body? Because muscle does not have glucose 6 phosphatase. The muscle can make glycogen into G1P, and that can be converted into G6P. But it cannot take the G6P and free the glucose. So muscle glycogen is only for its own use. The liver, by contrast, has glucose 6 phosphatase and can release the glucose into blood. The muscle can't because the muscle doesn't have G6 phosphatase, right? The muscle can take the glycogen. It takes the glycogen, it converts it into G1P and then converts it into G6P, right? And finally, can fat make glucose? This is a classic trap, right? Now, fat cannot really, not all fats can make glucose. Now, glycerol can get converted into DHAP, and that DHAP can go into gluconeogenesis and make glucose. But even chain fatty acids cannot enter gluconeogenesis, right? Um, Acetyl CoA cannot produce net glucose in humans. Fat provides the energy for gluconeogenesis, but ordinary even chain fatty acids do not provide net carbon for glucose. That's a very, very important distinction. So fats do provide the energy for gluconeogenesis, but ordinary even chain fatty acids do not provide net carbon for glucose. What I want you to remember is basically the big picture. So in fasting, the insulin is dropping and the glucagon is rising, and the adipose is breaking fat. And once the adipose is basically breaking fat, the fatty acids are providing energy, and the liver starts gluconeogenesis. The liver collects the lactate, alanine, glycerol. Can you see here? It takes all this and starts gluconeogenesis. The only pathway you need to remember in your head is basically the pyruvate carboxylase, the oxaloacetate, the PEP carboxykinase, and then we need to remember this fructose 1,6 bisphosphatase, which was being inhibited by AMP, and then we had the glucose 6 phosphatase, which was absent in von Gierke disease. I don't know why I've written von Gierke disease. This is so funny. This is supposed to be von Gierke. I'll stop the video here because I've said all I have to say for gluconeogenesis. I hope you guys understand.